Good? Okay. Welcome everyone to come here for the symposium. I know some of you fly a long flight like Rob, people from Europe, I don't know how many hours. And welcome all the people actually, as Martin said, in the past week for the summer school, we run pretty good summer school. And today this symposium is like continued education to show more, about how to use modeling to do immunology research. And show my slide. Yeah, where's the? Oops. Screen. Okay. My talk's supposed to be the last talk, suppose after you hear my talk, you can go home. But the thing, Fiona cannot make it, she lost her voice, since yesterday, she cannot make it. So I just replaced her for the first talk. And so, as Martin said, what I'm going to do is I will give you a quite brief overview about the research in our center. And basically what our center is doing is we are doing the computational or system biology research for immune response to influenza vaccine and also infection. And basically what system biology approach is, probably most of you know what the definition is. I think the system biology is supposed, the system biology approach, we're supposed to study the interactions between the components of a biological system. And also we're supposed to use a new perspective, integration instead of reduction. Traditional way is the reduction. And what that means is usually, traditionally when people do experiments, each lab only focus on one type of cell, even one, several genes you are interested or several proteins you are interested. Then now when we do the, system biology approach, we're supposed to integrate all the components at different levels together and to study the whole system. So that's the, different idea, different perspective. In terms of experiments, in order to do this kind of system biology, in order to use this system biology approach to do, investigate a biological system, then we need to measure actually not all, but at least most important components simultaneously at a system. So that's the thing. We need to measure many things at the same time. And also, for this system biology approach, this system biology approach starts because of the new technologies. So in particular, those different kind of high throughput omics technologies. So that means that give us a lot of data for us to look at a biological system in a more systematic way. So that's the thing. And also, for system biology approach, require rigorous data integration with different kind of mathematical models, computational models, and statistical approaches. So this is the different way to do biomedical research. Actually, when you talk about the system biology approach, actually there's a lot of challenges and a lot of new things we have to adjust ourselves to deal with it. One thing is, like I mentioned earlier, when we deal with the whole system, there are different kind of levels and also the data are quite complicated. There are so-called complex high throughput data at multi-scale, multi-levels, like genetic level, protein level, cellular level, organ level, even population levels. So how to deal with this kind of complex data, that's, this is quite challenging. The second thing is the relationship between all different kind of factors, at different levels are quite complex also. Usually when we do the traditional way, we expect the relationship usually is linear. When you see this is increase, that one increase, also increase, or this increase, that one decrease, or one, stimulate another one. That's the traditional way. We think that everything is kind of linear. But when we look at a biological system in a system biology approach, the relationship can be very complicated. Can be nonlinear. Even we have feedback effects. All those different kind of the relationships that we need to look at. Another feature for the system biology is that is dynamics. All the system actually is dynamic. So when we talk about the immune system, when our immune system try to respond to infection, there's a time factor. The immune system takes time to respond to the virus. Now, how to capture this kind of dynamic feature, that's also very challenging. What this means, when we look at the dynamics, the time evolution, or look at the time scale, then we need to measure things as a time course, not just at the steady state. So this is also very important. And also definitely when we do this system biology research, we need support from different kind of interdisciplinary, so-called interdisciplinary quantitative sciences, because not only statistics, not only mathematics, not only computer science, we need to integrate all these different kind of things, all the different kind of computational quantitative technologies to deal with the data, deal with the system. So that's why this thing, is quite complicated, quite sophisticated, When we talk about, more details, here in our center actually, when we start to use this system biology approach to work on our system, there are different kind of levels we have to think about this kind of system biology approach, at different stage also. The first stage, when we think about system biology is at the experimental design stage. At this stage, what that means, we need to, think about the first thing, as I mentioned, we need to think about integration idea. How, instead of traditionally, reduction. Basically what we need to think is, we need to consider the biological system as a complex dynamic system with a larger number of elements and components at genetic, protein, and cellular levels. Right now we are not dealing with more like a higher level, like organ or population level, but at least at 3 levels, genetic, protein, and cellular level, we need to think about the immune system as a system. So that's the first thing, when we try to design experiment, we need to think about. And the second thing, because we treat the immune system as a system, then we need to disturb the system from one steady state to a new steady state. When the immune system or any engineering system, is in a steady state, you cannot see anything. Basically, it's flat. Everything is flat. You cannot measure anything. In order to understand the dynamic system, then we need to disturb the system in some way. So here, we use virus or use vaccine to disturb the system and to let the system start to work. Then we can try to capture the dynamic feature of that system in order to understand that. Another thing is, definitely in order to design this well, and there's no standard way. You cannot do like a standard statistical design, calculate the sample size, how many subjects or how many data points. It's hard to do that, in a standard way. There's no software can do that. This is a very complex system. You cannot do this. Basically, in order to do this, we really need kind of like system modeling, simulation, and definitely complex statistical support to really design this, this kind of system biology, experiment well, efficient. So that's the approach, we are trying to deal with this. And the second step, definitely, once you design the experiment, you need to perform the experiments. So when you do the experiments, you need to keep in mind several things. So one thing is we are trying to measure all the key elements simultaneously. Definitely, it's hard to measure everything in a system, but at least we try to measure all the key elements we think is critical, and ideally it's at the same time. Sometimes we may not be able to do this, then we have to find a way to deal with this. And also we have to think about the latent variable or the limitations. Definitely some of the key elements, maybe we have the technology limitation we cannot measure. The question is how to deal with this. Usually we call that kind of unmeasured variable as a latent variable. Can we deal with this using a analysis approach after we did the collection? Then we have to think about this during the experiments also. And as I mentioned earlier also, we try to capture the dynamic transition period. When we design the time points, at what time to measure, we have to make sure before the system gets into the second steady state. Originally they have a steady state. You try to disturb the system, let that system start to work. Then you have to put your measurement points at the during the period that before the second steady state is reached. Otherwise, it's also flat. After another steady state is reached, then everything becomes flat. You cannot capture anything. So this is what we need to pay attention. And definitely another thing is, for this kind of system biology experiments is very expensive. As you know, because we are trying to measure, many things at the same time, as you know, definitely there's a lot of labor. You know, you need to involve many people doing this. You know, one lab may not be able to measure everything. You have to involve many labs, different kind of labs with different kind of technologies to do this. You know, more expensive, you know, cost in terms of cost and labor, you know, everything. So this is the thing we, you know, we are thinking in the design stage. Then after the experiment, during the experimental stage, you know, after the experiment, definitely we collect a lot of data, and then we need to think about, you know, data management, data analysis, and data modeling. You know, this is the, you know, another critical piece, you know, in our center is we develop, you know, novel technologies, novel methods, you know, statistical methods and mathematical methods to deal with the data. So in particular, the statistical methods to deal with the mechanism-based models, like you know, when we use differential equation models, maybe some others, you know, stochastic differential equation models, different kind of complex mathematical models. How to link the data to those models is critical. Usually there's no existing approach, statistical methods to deal with that. So in here, you know, we develop, you know, a lot of new methods to deal with that. And then definitely we need to think about this, you know, the system analysis and also predictions. This requires some kind of system engineering technology to deal with that. You know, finally, definitely we need the annotation and interpretation for the result. You know, after you play with the data, do a lot of mathematics, computing, and then we need to, you know, do a good annotation to make sure we really get a very important biological findings. This is also very challenging. You know, right now the data is, you know, is huge, and how to, you know, annotate the result, that's quite challenging, you know, to deal with. And we need bioinformatics software and database, and also we need to try to understand both biology and also the computational results. So that's, you know, that's the thing, you know, we need to gradually to learn and to work on. Now I will turn a little bit more on mathematics, statistics, because my talk is actually supposed to be the last talk in a statistical section, but you know, I will talk about a little bit more about mathematics, statistics. Even I move my talk to this first session here. And when we talk about mathematical modeling or computational modeling, there are three different types of models when people do. You know, in particular, those biomathematicians and or theoretical or biologists. When people develop mathematical models, there's three different things. You know, first thing is many people actually doing, you know, the computational models just based on the biological theory, and usually they don't have data or they don't use any data. You know, just to do the theoretical models. This is the first type of models. Second type of models, they develop some models based on the biological theory mechanism, with partial data validation. You know, after you develop a model, you just do simulation and compare your simulation result with some literature data and to see whether it's produced some kind of, you know, similar patterns. So that's another kind of a practice for modeling. The third way is with, you know, the mathematical models with rigorous data justification. So this needs statistics. You know, when you really rigorously link your data to the mathematical models, you know, that's, you know, really need advanced statistics. This is what we prefer. You know, we try to practice here in Rochester. You know, so the question is not only to see, create some mathematical models based on the theory and do simulation, you know, to show, oh, my simulation result, you know, have a similar pattern to the data somebody else published. That's not in that way, but we really expect to use our experimental data and to validate the model, and also hopefully this model can do good predictions for the future. So that's the direction we go. And when we talk about the mathematical models, there are different type of models. You know, different people use different kind of models. You know, particularly, you know, the differential equation model, different kind of differential equation models are very popular. Like usually, you know, ordinary differential equation models, such as ODE, or delayed differential equation models, or partial differential equation models, or stochastic differential equation models. These different type of differential equation models, you know, are very popular in modeling biological process, because this is very natural. You know, when you use, you know, differential equation models to model something which is a dynamic system, because the dynamic system is supposed to have time involved. So when you use differential equation models, you know, the time can be automatically, you know, naturally incorporated. You know, and actually here, you know, in Rochester, we focus on the first one. The first one is the most simplest differential equation model, so ordinary differential equation model. We do a lot of ordinary differential equation models. What we are thinking is, if we cannot do the simple thing, we cannot do more complicated. We want to start from the simple model first, then we can move on to do more and more complicated models. And some other type of model, like state space models, and that depend on, what kind of state space models. When you do the like engineering type of state space models, this kind of model is the kind of discrete time point models. And also people probably do like stochastic process models, like branching process or some other kind of, the stochastic process models, also involve time. This is also good to deal with the time. And also agent-based models, like on Wednesday we have a whole day summer school talk about agent-based models. Many people doing the agent-based models try to model immune response, system. And also the network models, different kind of network models are very popular to deal with like signal pathways of gene regulatory networks and metabolic networks, etc. So there are maybe some other different kind of models. So this is the model, usually people do. Today I'm going to focus on the first one. Again, it's the simplest one, the ODE model. So when we talk about this mathematical modeling approach, there are two modeling problems. The first one is so-called forward problem, and the second one is called the inverse problem. The forward problem means given the model and the model parameters, how to use the model to do simulations, predictions. So in order to do this, if you know mathematics and computing, probably you can do this. Okay, this is the forward problem. The model is established. You just use the model to do prediction and simulations. Okay, and the inverse problem is given the data from the model, suppose, and how to identify the model structure, how to estimate the model parameters. Model structure, everything is unknown. How to identify this. So this is the so-called inverse problem. Because this involves data, that's why, we need statistical methods to deal with this inverse problem. Okay. And basically from my perspective, I think the inverse problem is much more challenging, more difficult than the forward problem. That's why, the forward problem right now, the computer is so powerful, so you can easily do, the simulation prediction. It doesn't matter how complicated your model is. But for inverse problem, it's so challenging. Why is that? I gave a reason for this. The first one is, in order to do the inverse problem, you have to solve the forward problem first. Okay, without solving the first, forward problem, it's difficult to do inverse problem. And also, there's an identifiability problem for complex mathematical models. Even for differential equation model, ordinary differential equation models, if you write down a model, I give you a bunch of data tell you, for this variable I can measure for these 10 points. How do you know whether the model parameter can be uniquely identified? You can probably use a different set of parameters to do simulation to fit your data very well, or can simulate the result are similar to the data. Even the parameters are different. So that's possible. So the question is how to deal with the identifiability problem. You really know this, you really can uniquely identify your system, your model, and your parameter. So this is the challenging. And also, usually we have a high-dimensional parameter space. When we deal with in particular at the protein genetic level, there are many variables, many equations even. That means that you need estimate a lot of parameters. But how to deal with the high-dimensional parameter space estimation problem? That's the thing. And also high computational cost. So high computational cost because you need, in order to do the inverse problem, you need to repeatedly solve your equation many times in order to do the optimization, to deal with the parameter estimation. So this usually, you need a probably very powerful computer to deal with this. And also the model structure uncertainty and identification is always a problem. If you know the model structure, it's linear, nonlinear, that's kind of easier. But even you don't know what model structure looks like, that's another layer of complexity to deal with. And also, some of the parameters in the equation may not be a constant during your dynamic process. Like I just mentioned, the whole, the system is a dynamic. During this dynamic period, maybe some of the parameters may be changing. How to deal with that? And also the data, problem. Data always sparse and noisy from the biological experiments. And, even we are talking about like high throughput omics data, I think, oh, we got a huge amount of data. However, those huge amount of data actually for many genes or many proteins, compared to the number of genes and proteins, the data are still sparse, even if it got huge, right? So it depends on what do you mean, sparse and intensive. It's intensive in total, but if you in average per gene or per protein, whatever, that data still sparse. How to use the sparse data to estimate the whole system. So that is challenging. So and also how do you evaluate your model is right or wrong, whether it's how to deal with the model evaluation, diagnostics. So this is also the issue, new approach we need to develop. So all these issues tell us that the inverse problem is complicated. And however, when we use the system biology approach to study the immune response to flu infection or flu vaccination, we have to deal with all these kind of inverse problems. Otherwise, you cannot identify your system. You just look at your data without looking at your system. There's, you still don't understand the mechanism behind it. If you really want to use mathematical models to understand the whole mechanism behind this, behind the immune response, you really need to solve all this. So this is the research, that we are really, focused on in the past several years, actually, in our modeling, group. And then let's talk about, real mathematics, okay? And so here is some, the very simple, not very simple, but for some people are very simple, and ordinary differential equation model. Usually when we talk about ordinary differential equation model, we mean, the variable x, derivative of a variable x equal to a function of this, maybe this f is linear or nonlinear, a function of f and xt, maybe also have involved t, and the theta is the parameters, it's unknown parameters usually. And this is a starting point, starting condition for this differential equation model. For this equation model, for this differential equation model, the x basically is the vector that describe doesn't matter you are looking at cellular level, protein level, or genetic level, factors. If it's cellular level, this is like probably cell concentrations. If this is genetic level, maybe these are gene expressions, for different genes. So, so this is the system we are trying to investigate. We try to understand. Although in particular, we try to understand what is the structure F is, what F looks like. And also we try to estimate what this theta is equal to 1 or 2, 3, how to estimate this parameter theta. And in order to do this, we need to do experiments to measure something. Without data, we cannot do this. So basically what we measure is y, which is a function of this x, this vector, whatever you're looking at, cellular level, genetic level, protein level. So plus a measurement error. This is a measurement error. So question is, how can we use this data, the measured data y, try to do inference or try to understand, to identify the structure of f and also the parameter theta. Even sometimes this measurement equation also has some kind of unknown parameters we need to estimate. So this is the challenging question, okay? So usually for the differential equation model, if f is a nonlinear function, we don't have a closed form solution to this. In order to solve this differential equation, we have to use the computer numerically do it, okay? So the question is, when we numerically do this, to solve this differential equation, how do you know which parameter of this theta? Even you know the structure first. And how do you know which parameter can be uniquely identified based on the measured data you have in your hand? So this is a so-called identifiability problem, okay? So even for this simple ODE model, differential equation model, this question is not that easy to answer. And so far, we work on this problem, many years ago, not many years ago, probably 4 or 5 years ago. We just published this review paper in the last year, in the SAM review. And this paper, reviewed a different kind of methodology to study how to study the identifiability problem for the ordinary differential equation models. What that means is we can use some mathematical tool to tell you, if you give me your data, I can tell you which parameter can be identified, which one cannot. Okay, that's the technology we have. I don't have time to talk about this in details, but there are some technologies. Even this technology right now, we are only able to deal with probably 3 up to 5 equations. More than that, it's hard. Even whatever the methodology you use, probably the simulation method is okay, but mathematically it's difficult to deal with more than 5 equations, probably more than 10 parameters. Fewer than 10 parameters we can do this, but otherwise it's hard. Okay, and then if you know your parameter can be uniquely identified, then the question is how do you estimate the parameter using the data? Because you don't have closed form solution. This is not a standard statistical question because this is not a regression. This is a different equation. You don't have closed form. How can you use this data Y to estimate this parameter? Let's right now assume this F is given, is known. You know the structure of the model. I will talk about if you don't know the structure of F, we still can do it. Also can do it, but less cost more data. And but if for given F, how to deal with the parameter set, how to estimate the parameter set. There are several different kind of methods. Most of the mathematicians probably know the least square method. Standard nonlinear least square. You just use brute force, try to minimize the least square principle. Then you can try to estimate the parameters. In order to do this, you need to numerically solve the differential equation many times in order to minimize the least square objective function. And usually for this thing, if you deal with 3 to 5 equations, probably 6 to 8 parameters probably are doable, but it still take a lot of effort to make it work. The difficulty is you have convergence. You always have convergence problem. Most of the cases, the differential equations are nonlinear. So the local minimum is a problem. Sometimes you get a result, you don't know whether it's local, it's global, whether it's the best solution. So, and the computational is very expensive. When you deal with the nonlinear differential equation, you need to solve it many times to deal with the least square estimation. That's always is a problem. So. And then our group, and maybe with others also, we developed some new methods to deal with the computational issues, using so-called two-stage smoothing approach. We'll talk about this later, a little bit. So this one can give you a very fast, very quick result, without convergence problem, but you have to pay. The price you pay is the estimation accuracy will be lower than the standard least square. So that's, there's nothing is free, okay? Then there's a, Ramsey's group from Canada developed a method called penalized smoothing or profiling approach to estimate the nonlinear differential equation models. And also there's a Bayesian approach, try to use the prior, because sometimes it's difficult to identify the parameter, then you have to borrow some prior information from others. And also I will talk about later, about how to deal with the structure identification. If you don't know what the function looks like, how to deal with that. So I will talk about later. Then before I go to more like a statistics, and I will give you an overview, very brief overview for what we have done in our center in the past several years. Basically our center called Center for Biodefense Immune Modeling, and the goal is we try to develop mathematical computational models for immune response to influenza infection and vaccination based on experimental data from mice and human. So what that means we have to do experiments for mice and human, and collect a lot of data, then to try to establish those mathematical models. So that's the goal. What we are doing. Another goal is that we need to develop user-friendly modeling tools and software packages, to share with others, with the general research community. That's the goal. What we are doing. The people here, basically, this is really interdisciplinary, project. So there we have different kind of people. And like we have our immunology core, like Martin's heading the immunology core. We have 1, 2, 3, 4, 5, 6, 7, 8, probably eight labs. Each of these labs, is trying to use their technology to measure different kind of things. Like David Topman do the CD4 T cell, also is good at mice experiments. Steve Wiley is our genomics, center co-director to help us to get those gene expression data, RNA signal, microarray data. John Trainer is a clinical investigator. Tim Mossman do the CD4 T cells for us, and Alexandra Livingstone do the dendritic cells. Brian Ward doing the virus, and Martinez also develop new assays for us to measure the viral titer. And so this is they involve many immunology labs, with different kind of expertise for this project. And I don't have space to list all their postdocs, technicians who spend a lot of time and labor to collect all this data. Really, they work very hard to do this. This is the experimental people. And from our quantitative science people, we have here bioinformatician and statistician, mathematical modeler, and computing people, and also we have data management and software development team. And definitely we need IT support, hardware, software, IT support. So all these different people, we work together as a team, to deal with the complex system, because what we are dealing with is the complex immune system. So our team also is a complex system, this complex system deal with this system. I put all these arrows, you probably cannot see clearly. This is how we interact with each other, different kind of people here, to interact, to work on the system. Otherwise, it's not easy to get it. So that's involve many different kind of people to do the work, actually. So I will show you a couple of, a project we have done in the past year, we already published. This is the first project, we did is published in Journal of Virology in 2009. So this paper is talk about simulation prediction of the adaptive immune response to influenza A virus infection. This is a typical paper that belongs to type 2 models. What that means? That means this is the theoretical mathematical models with very limited data, not validation, just to compare to the data. And basically, based on all the biological theory, what we try to build here is we build a mathematical model for the flu infection in mice. And basically, what we are looking at is the flu virus can infect the target cell, which is on the epithelial cells in the lung and the airway, and turn this into infect cells. At the same time, the flu virus can stimulate the dendritic cells or infect the dendritic cells, and dendritic cell traffic to the spleen and lymph node to stimulate the immune response. Basically, the dendritic cell can stimulate the CD8 T cell response and also CD4 cells, and also stimulate the B cells, and the B cell will produce will turn into the differentiate into the plasma cells, and the plasma cells will produce antibody. That antibody will kill the flu virus. At the same time, the CD8 T cell also kill the infect cells. So this is the two arms of the immune defense. We put all these components there, about 15 of them. And when we, I'm not sure I have the equation. I don't have the equation. Basically, for each box, it's like one differential equation. If you put it together, this is the 15 boxes, and then we have 15 equations. And in this 15 equations, we have like more than 60 parameters. It's quite complex. So basically, it's hard to, as I mentioned, it's difficult to estimate all the 60 parameters in the 15 equations. So now we have to use some other strategy to deal with that. Our strategy, actually, we try to disconnect each of these, some of this link to reduce this to different blocks. Then we try to estimate parameters block by block, block by block. Then after we build the block, then we put them together again, try to collaborate. So this is kind of the strategy we are dealing with. And so again, when we do the simulation, this is the type of model. We can write down the model. We can assume some numbers for each of the 60 parameters. We can do simulation. This is like a forward problem, right? We can do a lot of simulations. Then we grab some data. This data is the data from some literature. I don't know. Even the data is probably from some of the data from mice, some data from the human. Or, we don't care too much. So here is the typical. The type 2 models. You just do simulations, then just put your simulation result. This curve is a simulation result to see whether it's kind of like produce similar patterns, to the data, you collect from the literature. That's not much, just some sparse data over there. Like this is the data for virus, this is the data for dendritic cells, this is data for CD8 T cells, and this is the data for antibody. Some of this, sparse data we try to see, okay, our model can produce similar patterns to those real data. But you don't know how those data produced, under what condition, you don't know. So, but this is just type of the thing. And then this is the second example. This is the paper we also published in Journal of Virology 2010. So this paper actually, we are doing the type 3 models. This one is really we develop a model and rigorously use our, do design experiment, collect data, and then use data to rigorously identify the model. So this is the, the one of the practice. Again, this is the data from mice infected with flu virus. And so the model basically is 3 equations. This is here we, as I mentioned, we cut those 15 box network into a block. This is one block, which is the long compartment. We just consider the epithelial cells, infected epithelial cells, and the virus. Okay, so this is the 3 equations. In these 3 equations, we only have 6 or 7 parameters. I didn't count it. Probably 1, 2, 3, 4, 5, 6. I mean, plus these 2, 7, 8 parameters. In this, 3 equations, what we can measure is the viral load. And this is the CD8 T cell count we can measure, and this is the antibody we can measure. This is a different kind of IgG and IgM antibody. Many of the modelers know this kind of standard, the viral dynamic models, and then we try to see answer, to ask a question. We only be able to measure this virus. We cannot measure epithelial cells and infect epithelial cells. Can we identify all the parameters from this data of virus? So let's assume the data has no error, perfect. You can get whatever infinite number of that data. Can you do that? Theoretically, the mathematical analysis tell us that all the parameters can be identified except this one. I put a different color. So this one is not identifiable. This confounding with the initial value of this target cell concentration. So, but the interesting result is even this one cannot be identified. You can fix as 1, 100, even 1,000. It doesn't matter. Theoretically, we can prove, from simulation, we can prove whatever number you fix this will not affect the estimate of the rest of other parameters. So this is a good property for this model. So that's what we did. This is the data we collect. This is the virus titer, data for after the infection of the mice. We collect the data from time 0, day 0 to up to day 14. So the very frequent, a lot of replicates at each time point. This is the data, this is the amount of data we need to identify all the parameters. So usually people don't do this frequent data. So this is the CD8 T cell data, this is the IgG antibody, this is the IgM antibody. So you can see all the data are very frequent, and the variation is big at each time point, a lot of replicates, and this is how tell us this is the real pattern instead of everything is noise. Even if you look at the variation, it's large. Now we fit the data, this is the model fitting, results. The curve, is pretty, fitting pretty good for the viral load. So I don't have much time to talk about this. If you are interested in the scientific result, you can look at the paper. We do have the paper already published. So this is another paper we published. I will be very brief for this. This is a look at that CD8 T cell trafficking model. So this is another block in that big network model. So this also we use, we are looking at the CD8 T cells trafficking from lymph node, spleen, and the lung. How they traffic. So this is also 3 different, 3 equations. Then also try to estimate parameters. We also collect a lot of data for this. This is like a CD8 T cell data in the lymph node. This is data in the spleen, and this is in the lung, and this is dendritic cells. Okay. So then, we fit all this model, we estimate all the parameters, again, similarly as the viral dynamic model. So, this is the fitting result. It's pretty good. And again, I don't have time to talk about the science. I suppose only talk about statistics, but I just show you some of the data. We are still working on several other blocks, like CD4 T cells and the B cell, antibody production model. And also, after, as I mentioned, after we finish this, we're supposed to, integrate all these different kind of cellular level models into one system. So this is the integration, for this is the lung, lymph node, spleen, different compartments, and then we'll have different kind of, the type of cells, immune cells, and virus, to integrate. Finally, hopefully we can integrate them together, after we finish each block. Okay. Now I only have probably 10 minutes, 10 to 15 minutes to talk about the genetic level, and so we are, besides those cellular level, as I mentioned earlier, we are also looking at the protein and the genetic level and other levels. We don't have a protein level data yet, but we have a lot of genetic level data. I will talk about this a little bit. For the genetic level data, we are still trying to use the differential equation models, because if we want to link them together with the cellular level data in a differential equation model format, then ideally we should also do the differential equation model for the genetic level. So that's why we are trying to use differential equation models to model the gene regulatory network. Basically, this is like a kind of like a high-dimensional differential equation models. If we are looking at 2,000 genes, that means we are looking at 2,000 differential equations. Okay, and if usually you don't know the model structure. For the cellular level, you know the biology, you can somehow figure out some of the biological, some of the mathematical structure. But for the genetic level, you don't know each gene how they interact. So we have to identify the model structure. Okay, and also, the structure is potentially nonlinear and complicated. Their relationship, the gene regulation relationship may be very complicated. Again, the data is sparse and noisy, and also we need to deal with the high-dimensional nonlinear optimization problem, and also we do have computational problems to deal with, and a theoretical justification for the results. That's all the issues we are trying to deal with, and. Also, why we need to use ODE, to deal with the gene regulatory network? As I mentioned earlier, one is because we already done the cellular level differential equation model. It's good to continue to use this for the genetic level, and also, as I mentioned earlier, also, this ODE model is good to capture the dynamic feature of the gene regulatory network. And also, the ODE model is a directional graph. You have directions. Which one is a causal. There's a causal effect. Which one is on the left-hand side, which one is on the right-hand side. Basically, the right-hand side, gene will affect the left-hand side. So you do have a direction. So, and also can deal with positive and negative effects, and also feedback effects. And also easy for predictions. Once you have a model, differential equation model, it's easy to do simulation prediction. So that's a good feature for differential equation models. And when we talk about the differential equation model, let's do a simple one before we do the even complicated differential equation. The simple differential equation model. Let's say we assume we know the model is linear. Everything is linear, simple. Let's say the S gene, the derivative of the S gene's expression, can be regulated by the rest of the other genes in the network in a linear way. The linear way, basically, this Xj multiplied by the regulation effect theta. Okay, so but for each of these genes may be only regulated by several other genes. Not every, if we are looking at the 2,000 genes in the network, not all these 2,000 genes have an effect on one of the other gene. Maybe only 2 or 3. What that means is most of these theta, these parameters are 0. Only several of them are non-zero. So the question is, how do we know which one is 0, which one is not? So that's the network construction. Basically, I want to see for the S gene, I want to see which of the other genes in the network have a regulation on it. So this is the question we need to figure out. How do we know whether theta is 0 or not? If it's not a 0, what is it? Is 1, is 2, is 3, or how strong is the regulation? That tells us how strong the regulation is. So this is the problem we need to deal with. And definitely, as I mentioned, how do you know that's linear? If it's not linear, then we can do nonlinear. Basically, what nonlinear is, you can do any kind of nonlinear. If you know the form of nonlinear, for example, if the differential equation is nonlinear, like in this kind of form, it's kind of like kind of like a Hill function, c over 1 plus exponential or something. So this is like if you know the form, then you can write down the nonlinear form. Then you need to deal with whether this thing is 0 or not. You still need to estimate all the parameters. And sometimes, as I mentioned, we don't know the form. Then we need to write down the regulation effect as just a general F. It's unknown structure. Then we need to estimate what this F is. This is non-parametric. We don't know what form it is. We can estimate. This is like everything is additive. Looks like all the regulation effect is additive because everything is summation. So how to deal with the co-regulation? Okay, maybe 2 or 3 genes co-regulate the one gene.
So in order to deal with the co-regulation, we need to deal with besides this additive effect, we need to estimate this like co-regulation effect. 2 by 2 or 3 by 3, even more regulation. So this is the challenging question we need to deal with. How to identify all these model structure and also model parameters. So this is the thing we are working on. So one paper we just published, in JASA, this is the statistical journal, and this is for the linear model. If we know how to deal with the linear model, then we try to simplify all other complicated model into linear model, then everything is done. So this, for the linear model, we develop a 6-step procedure to deal with that. The first step basically is like a screening significant gene expression curves. Because right now, when we do differential equation, we have to measure all the gene expression in a time course, right? As I mentioned earlier, no time course data, we cannot identify differential equations. I don't have much time. And then second step is we try to cluster the genes into functional modules. In this way, we can try to reduce the dimension. This is dimension reduction approach. If you cannot deal with 2,000 genes, let's do the clustering, because many genes, they perform very similarly in a process. You cannot identify them. They just follow a similar pattern. They're very similar. So we can cluster those similar genes together so that at the same time you can reduce the dimension. And step 3 is then we need to smooth those data for each of the functional forms to estimate the curve, at the same time to estimate the derivative of those curves. Why derivative? Because differential equation left-hand side is derivative. So we estimate both population curve and derivative. Then we use this derivative estimate and also curve estimate into this step 4, to try to identify which parameter 0, which gene is connected with which. That's the step 4. And step 5 is just a kind of like a refinement for the parameter estimation or the regulation effect estimation. Because when we do the variable selection or try to identify which gene regulates which, we sacrifice something. Then here we try to get back that effect. Try to refine the parameter estimation. Finally, we need to do the functional enrichment analysis for the annotation for the network. So this is the 6-step approach. And so the key step again is we try to estimate each of the functional modules or each of the genes, their curve and also their derivative. Then plug these two things in the differential equation model, turn this into a regression model. Basically on the left-hand side, this is the derivative. So, but if we have the derivative estimate, we plug this in, then turn this into data. And the left-hand side basically is the original curve of the gene. So when we plug this in, this model becomes a standard regression model. Then we can apply the standard regression model selection approach to identify which of these parameters 0, which one is not. If it's not 0, that means that gene connected with this gene. So this is the standard like lasso scatter approach, quite popular in statistical field, and then we can do this. Okay, so this is the thing. Then before we apply our methods to our data, then we need to validate the methods. So we first use the very popular yeast cell cycle data to validate the methods. This 1, 2, 3, 4, 5, 6 steps. Basically for this yeast cell cycle data, there are 800 significant genes we identified from first step. Then we cluster these 800 genes into 41 modules. Then we do smoothing and then try to identify the regulation effect. Then estimation refinement and then enrichment analysis. So we go through all these 6 steps, try to make sure this really works fine. So this looks. Let me see my time. Still okay, have some time. So this is each of the yeast cell cycle data. The dot is the data, the curve is our smoothing estimate, and also the differential equation model fit. They fit very well. For each of the functional module, these multiple genes are there. You can see all these genes within each of the module, they are quite similar in terms of the time. This is just from time 0 up to this time, I think 2 cell cycles. I cannot remember exactly, maybe 2 hours or something. So during this time period, you can see within each of the module, all these genes, different genes, even multiple genes, they perform very, similarly. But for different modules, they are different. The patterns are different. Then we fit we smooth this data and also fit the differential equation model. Try to link which module reacted which. Finally, actually, we got all these 41 modules. Then we, based on our models, based on our different equation modeling, then we get this network. Basically this network is, is a direct graph. This centered modules actually is a hot top, regulate all, most of the other gene, modules. And so this, this is the way, we have to validate this methodology and to the existing data. Then we can try to apply this approach to our own data. That's what we are working on. We do have, our own data, like the gene, I just mentioned that for the mice experiment, we do have time-course microarray gene expression data. All this data, this is a cluster data results. It's quite good for each of the module. It's pretty consistent within the module, between module, they are different. And also we have the RNA-seq gene expression data from human subjects with flu vaccination. We got all this, data there. I don't have time to talk about this. Actually, we don't have a final result from this own data yet. After we validated those methodology, we are, working on this data right now. Hopefully, next time we can report the result from this. Finally, our final goal is from our center is we try to, we got those, cellular level models. At this, this is the organ level, this is the cellular level, this is the protein level, this is the genetic level. We got those cellular level models already. We are working on the genetic level model. We are going to do the protein level. Hopefully, we can put all these things together after we finish each of these levels, and also vertically we can link them together to make a whole system. So that's the system biology approach, what we are trying to do. So we do this step by step, even within each of the level, we also try to make it into blocks, and then put them together, integrate them together. Then finally we integrate all this together in a systematic way. So that's the way we are dealing with, and I don't have time to talk about this. And also we have a software, we have a poster over there if you are interested for this differential equation modeling software. One is called DDiscover for the standard differential equation modeling simulation and estimation. And also we have another software, DNetWeaver, for the network model. This is dealing with the high-dimensional differential equation model for gene regulatory network. So this is the thing we are dealing with. Finally, this is my final slide. And conclusion is, the computational system biology approach really requires an interdisciplinary team. One person cannot do it, I'll show you. And number two is very challenging to integrate experimental sciences and all different kind of quantitative computational sciences. You need statistics, mathematics, computer science, all those things. And so this quantitative science I list here, what do we need to use? Mathematics, computing, statistics, engineering, physics, bioinformatics, whatever, you do. And also, I want to emphasize this mathematical computational modeling is really useful to extract, more information from the, limited but very complex experimental data. So hopefully more and more people start to think about it in this way and to use this approach in the real world. Again, this is the team of our center. I really want to acknowledge. I don't have time to talk for each of these people, but most of the people here, if you have the opportunity to talk to them, you can talk to them here to look at, to find out more details for each of these pieces. Okay, I think that's all I have. Let's see. Any question?
Why don't I start out? So, you presented a framework for reducing gene expressions to similar time frame clusters.
Right.
And then treating those clusters as a method of dimensional reduction, so that you can model the time course behavior of those clusters, assuming that the genes in those clusters have somewhat similar function or connections.
Right.
But we know that things go up and down at the same point and may not, and may be assigned to a cluster, but may have very different linkages. Once you create this model, how do you trace back to individual gene pathways to validate?
Yeah, that's what we need to do the annotation from the database. The problem is we cluster this not only for the dimension reduction. Even within this cluster, you can all these genes. Each curve is one gene. All these genes, you cannot distinguish them. If you model this as each of one of equation, all these equations are not, they are the same, basically. They follow the same pattern. So then we have to do this. This is one thing, we have no choice. If you put this, each of these curves as one equation, this equation is exactly the same almost. So, when you try to do the annotation, I don't know, I even, like you blind me sometimes. Martin do the experiment, he blind me, he doesn't tell me what gene is this, each of this. Then, as an immunologist, you have to probably, or bioinformatician probably can help us to annotate, why this gene in one cluster. If this cluster regulate this cluster, why is that? You can go back to the whole group. At the same time, you can go back to each individual gene to look at why it's there. So, there's a lot of biology that we need to do annotation. Yeah, I agree. But not that easy, I think. Yeah, Catherine?
wiping out some of the characteristics of your curve without realizing. So if something is, for example, peaking very early in an experiment and very late in another replicate, then you're actually going to average the behavior in a number of curves that represents neither.
Yeah. Actually, when we do this smoothing, each of, I'm not sure you can see this, each of this cluster, we have a red curve, which is the population mean of this group. And actually, we use the so-called mixed effect non-parametric approach to smooth this. What that means, when we smooth this, we do get the population curve estimate. At the same time, we do get individual curve smoothing estimate. So we do get both, And when we do this, actually, when we get the individual curve estimate, at the same time, we borrow information across this group. If without borrowing that individual curve, you don't have that. Again, individual curve data are sparse. So you may miss something, or you may, when you do the sparse smoothing, it's hard. So that's why we do the non-parametric mixed effect model approach. You do get both, the population curve smoothing and also individual curve. If you want to look at individual curve feature, you can. Yeah, that's what we actually, after we do the smoothing, we look at the individual curve to make sure each of the individual curve get into the right group. Sometimes somehow we may misclassify them. Yeah, that we did that. Yeah.
Do the clusters that you find, do they make sense biologically? Do related genes end up in similar clusters?
Actually, we did the annotation for this. Yeah, we got a lot of biological annotation for each of these groups. It's hard to see they are similar, they perform similar functions. When you use the GO to do the annotation, they tell you, this group, this bunch of genes perform mainly this 1, 2, 3, 4 functions which are significant from this. Yeah, we do get all these annotations results. But you cannot say all these genes are biologically or whatever, they are same. But you just do, that's different. Yeah. Any other? Yeah. Mm-hmm. We haven't, for our data, we haven't yet, because as I mentioned, right now we just, at the stage, we are doing each levels. We are not linked between levels yet. So we are trying to make sure each level we can do it well, then we can find a way to link this genetic level to the cellular level, So right now we're just still working on each individual levels. We haven't finished the individual levels yet. Yeah.
You said you had 41 clusters. How did you decide on the number 41?
Yeah, this is a good question. It's hard. And actually, there's no rule. I mean, even there are some rules, but none of those rules works actually. As you know, you want to do clustering. There's ASE, BSE, whatever those rules you use. There's nothing you can say, I should use 41 cluster or 42, 43. There's no rules. This is really a kind of ad hoc. You have to stop somewhere. You think you feel comfortable. So that's the thing. I mean, for all those clusters, you always have this kind of issues, unless there's something, there's a clear cut. Usually it's not. Yes.
Related question on the choosing the numbers. So also, even though you have sparsity constraints in there, it looks like your network is very, very highly connected. How do you?
You mean that example, that East data, right? Yeah, this one. Yeah.
So that's something critical. How do you?
Yeah, we can. If you don't like the heavy connection, you can tune in your parameter there to make it more sparse.
And why did you decide on this one?
This one is based on a statistical rule. We stop here based on there's a statistical rule like BIC to select where you stop, how many edges you get. Yeah, that there is tuning parameter. Again, there's no clear cut threshold, right? You can tune in that parameter. Oh, I don't like each of like in average, I don't like the connection more than 3. You can do that. It's here. I think for this network, I think average connection for each module is, I don't remember, 4 or 5. So if I don't like more than 3, then you can do that. You can tune in your system to do that. Yeah, this is the hard question. It's similar to the number of clusters. Where you stop. There's no. This is like when you do the standard statistics. You say my significance level at p-value 0.05. If you ask a statistician why do you choose p-value as 0.05 as significance, there's no reason, right? You just choose it 0.05. Why not 0.06? There's no argument. You just pick the one number, right? Yeah. Any other? Yeah.
Because several of these time courses are very similar, so you have very related clusters. I would expect that there's very many local optimized this data, that you can either regulate something with one cluster or with the other, something very little different. Do you have a clue on how many local optima there are, how unique is this solution?
Yeah, for the local optimization result, right? Yeah, when we do the optimization, whatever, when we construct this network model or try to estimate all those different equation parameters, there are many, many local minimum, and we are trying to use the global approach to do the optimization. There's like evolution differentiation, whatever, the different kind of like a genetic algorithm approach to global search the solution. Again, you can stop at some point. Even kind of like a brute force, but it's you have to run this many times, and also even you can start this from different starting points and then do many times, and finally you stop somewhere. So you think this is the best solution? Yeah, definitely. This is also, there's no good validation to tell you this is a true optimal best solution, but at least you stop somewhere. You look for the whole space. Yeah.
I think we need to move on. Thank you, Hulin.